Hi guys, so this video is going to be covering the cell cycle. Now the cell cycle, as you can see I've already drawn out, has four distinct steps to it. We all know about mitosis, and there's sub-phases for mitosis, so there's prophase, prometaphase, metaphase, anaphase, telophase. I'm not going to get into detail on those, but obviously you do have to know they exist. But we're going to dive in first with the gap phase following mitosis, and this is G1 or gap 1 phase. And this is the sort of recovery period for a cell once it's gone through telophase and separated into two distinct cells. Now the gap phase also has something else associated that's very important, which is the restriction point. And the restriction point is sort of controlled by retinoblastoma protein, which I'm abbreviating as RB. Now it's the point of no return because once a cell has gone past this point, it will divide. It will divide. And RB, retinoblastoma protein, is the only thing stopping it from happening. So, there are multiple levels of control here on retinoblastoma that I will get into, but it's just important to realize that this is the most important point of the cell cycle because it's the point of no return. Now, the S phase, or synthesis phase, is the phase in which we are replicating our DNA in preparation to start dividing again. And so this is where we go from a ploidy of 2N to 4N, and it's just sort of a copying pasting of all of our DNA in the cell. And then the gap 2 phase, the G2 phase here, is actually sort of the prep for mitosis. We are creating enzymes and proteins to help organize the cell, as well as checking errors that could have been made during the synthesis phase when we were replicating our DNA. Now, I've pointed out the most important step, the restriction point, but it's also worth pointing out that there are other checkpoints as well. But these checkpoints are different from the restriction point in that they are actually stimulatory, so I will draw them as arrows instead of a flat bar. And they are stimulatory to the cell cycle, so they are what give the cell cycle the directionality in this direction, from gap 1 to synthesis to gap 2, etc. So what's responsible for the sort of maintaining the checkpoints at these steps are cyclin-dependent kinases and their associated cyclin molecule. And I will abbreviate the cyclin-dependent kinase as CDK. And for this one, it's CDK2, and it's cyclin E. Now these are two separate molecules that act together in a complex, so you'll hear a lot the cyclin complex, I will say. And these two act together to sort of give the go-ahead for the cell to move from gap 1 to S phase. And over here, between S and G2, it's actually cyclin-dependent kinase 2 again, so it's CDK2 again. But instead of cyclin E, it's going to be cyclin A, so I'm just going to abbreviate CYCA. Then finally, up here between G2 and M, there is just one CDK, CDK1, but there's actually two different cyclins that can act here. So there's cyclin A or cyclin B. And it's frustrating because we don't know really any more details about these cyclin or complexes than that they act as the sort of gatekeepers to the rest of the cycle, but that's the level of detail that you're going to need to know about the cyclins. But about the checkpoints themselves, there is a lot more we have to cover. So, the most important point is that all of these checkpoints have their own associated inhibition markers as well. And these are what we call tumor suppressor genes. And in this case, both of these two checkpoints share the same tumor suppressor genes, specifically P27 and P21. And these are actually part of the KIP family, but you won't need to know that, the KIP family. And over here, this retinoblastoma step, well, we need something to inhibit retinoblastoma from functioning so that we can have cell cycle progress in the first place. So these are also a cyclin complex. So we have CDK4 and 6 this time. And that has its own cyclin associated, which is cyclin D. So that's an additional level of control. But on top of that, we have, again, these tumor suppressor genes that are functioning, that are also inhibitory. And so this is the P21 and the P27 that we're already familiar with, but also P16, which is actually the more important one that we care about. So these tumor suppressor genes will inhibit this cyclin complex and decrease its effectiveness such that retinoblastoma can continue blocking the cell cycle progress. And then there's one additional level of control even on top of this, which is simply another tumor suppressor gene that can interact with these tumor suppressor genes called P53. And P53 is normally complexed with another molecule to keep it inactive. But when there is cell damage, it will dissociate from this molecule, which is known as MDM2. Normally they are bound together, but when there is cell damage, so this will be my cell damage marker, they dissociate from one another, and P53 will stimulate these tumor suppressor genes, which will increase the amount of inhibition on the cyclin complex. The increased inhibition on the cyclin complex means that we have decreased inhibition on the retinoblastoma, which means more retinoblastoma, which means less cell cycle cell growth. So that's a lot to sort of take in all at once. So let's sort of go through it step by step so you can get a sense of what I'm talking about. So first off the bat, we have decreased MDM2. And hopefully I'm going to color code these as well so you guys get a sense of what's going on. So decrease of MDM2, and MDM2 is an inhibitor basically of P53. Now, the decrease in MDM2 because of cell damage will result in P53 being more available, so it's essentially an increase of P53. So it's an increase of P53, and P53 is stimulatory on these other tumor suppressor genes. So the increase of P53 will basically result in an increase of, I'm just going to write P16, and you can know that I do mean all three of these. But since it's the increase of something that's inhibiting an effect, I'm going to write it in blue. So you have an increase of something that's inhibiting, so you're going to have a decrease of the next thing as well. So that means you're going to have a decrease of the CDK4/6 complex. I'm just going to write CDK4/6. Decrease of CDK4/6. So that decrease of CDK4/6 is actually going to result in an increase of the retinoblastoma protein. Now, the increase of the retinoblastoma protein means that we have increased inhibition overall. So the increased inhibition overall means decreased passage through the cell cycle. So, what can we take from this? We realize then that when there is cell damage, P53 is expressed such that we decrease cell growth, which is what we want. We want to prevent mutations from causing uncontrolled cell growth, causing cancer, causing tumorigenesis. If we lose P53, however, if we have some mutation that affects the tumor suppressor gene, we will get a condition called Li-Fraumeni syndrome. And that's just, it will cause tumorigenesis. Because this whole pathway, we lose this ability to signal a cell to stop growing when we notice cell damage, say from ultraviolet light or something like that.